Maybe I can start the discussion by the last question. So one of the issues I think that clearly has changed in terms of safety signals post-approval is that, 20 years ago, when we got a drug approved through, after a successful phase 3 study, patients that were put on newly approved drugs were not nearly as heavily pretreated as they are today because we have many more drugs available. And so our selection for patients, or our selection criteria for patients who go on phase 3 trials are relatively stringent. When it gets out into marketing, we're treating patients that have been much more heavily pretreated than patients that were treated 20 years ago. And so I think it is true that our patients now live longer, therefore they've been treated with many more secondary and tertiary regimens. And so we are going to see more toxicity in a post-marketing setting than we did 20 years ago. I think that's just a reflection of where cancer treatment sits today.
If I could just add a comment to Lee's point. So I think that there is also another opportunity, both as you imagine the evolution of cancer in a patient as a natural consequence of progression and exposure to a variety of these drugs. The phase 1 patient is a difficult patient then to expose to a new drug. And the context has certainly changed. And so something we can do in comparative oncology is to allow those first exposures to occur in the context of minimal prior therapy. And so I think that is another opportunity, separate to the point that Lee was bringing up.
I have a question for the panel. Myrtle Davis, National Cancer Institute. I thought the data that you presented on osteosarcoma and metastasis were particularly provocative, and it looked like an interesting place to go. I do have a question about what you said about disease prevalence in the dog. And I often wonder if disease prevalence in the dog is factored into your value proposition because, again, just recently at ASCO, there's been some questioning about whether the disease itself actually matters with regard to therapy. So if that is indeed the case and the disease does matter, would you expect that the prevalence of disease in the dog, that might actually skew the opportunity quite a bit with regard to being able to test certain agents and their probability for efficacy in humans?
Yeah, so I think at one level, so the question is a strategic question about how feasible it is for you to enter patients into a trial. And so if you define disease histologically in osteosarcoma, we know that it's a very common disease in dogs, and then is an advantage relative to the rarity of that same disease in humans. If you were to, we can't yet say whether osteosarcoma in dogs is associated with ATRX dysregulation to the same extent that we could potentially say that in human. Osteosarcoma has been well studied, so I think the second part to your question is, if we start defining disease by molecular alterations, which we should, we don't yet know the prevalence of those molecular alterations. So the characterization is not complete, sufficient to answer those questions. So as Lee suggested, if we say we really want to study in the context of various histologies, KIT inhibitors, we don't yet know how many dog cancers are associated with KIT dysregulation. We know examples, but we don't know the breadth. So that I think is an opportunity or a gap.
Yeah, Carl Barrett, AstraZeneca Pharmaceuticals and formerly of the NCI. So the panel is a lot of my friends there, and obviously Sean knows I'm a big fan of the comparative oncology program, but
this is a bad sign.
But I did want to there were a lot of statements made this morning about success and failures in the pharmaceutical industry, and it was based on old data. And I just wanted to bring you up to date on at least some anecdotal data currently. So the study that Lee showed with 5 drugs approved with the 5% success rate should put it in context that there were 5 drugs approved in 2015, the 5 months this year. So there's well over 30 different targeted therapies and many more other therapies that have been improved in the last 10 years. So the success rate, I don't know what it is still changing, is rapidly changing. Second point is that we've actually have a drug that's been submitted for approval where the first patient was dosed 2 years ago. So the speed with which we can do this drug development, understanding the drug properties, understanding the right patient population, having the right molecule can all be greatly accelerated in humans. Third point is that we have presently an 80% clinical benefit response in phase 1 patients in our oncology portfolio. That's not to say that we have 80% of those drugs will go on to be targeted, will be approved drugs. Actually, I don't know what the percentage will be. Drug development in humans is hard. It's very hard. We have to do, as Mike said, combination therapies to really get durable clinical benefits. There's a lot of room for value for the comparative oncology program, but all models are wrong, some are useful, so let's focus on where are the useful parts. Thank you.
I completely agree, and that's why I tried to make the point that, phase 1 response rates are just completely different than they were 10 years ago. There's no question the paradigm is changing, and I didn't have the most recent data. I don't think we have it yet, but clearly we all expect this to be better. But I do think the real valley of death, the real attrition still from phase 2 to phase 3, we need to figure out how to make sure we minimize both cost and cost to patients, exposure to patients, and make sure we have less failures. As you know, that's the big investment once you go into a phase 3 study.
Yeah, for us, the biggest problem is really getting the patient population correct. And the biggest challenge I have with the canine approach is that you don't understand the genetics. You're a good 10 years behind where we were with the human, so that's where you have to catch up. The second one is the heterogeneity that Sean mentioned. So we actually, it's interesting, now that we have better ways of measuring quick responses, we can oftentimes show that we get very quick responses over 1 or 2 months, and those tumors keep roaring back. The approved drugs, they roar back after 12 months. But with other targeted therapies, they come back within 2 months. And that's all because of clonal selection for pre-existent resistant mutants. And we really need to understand that in the human and then figure out combination therapy. So, I think that's where we should be thinking about how the dog can actually have a value for human drug development.
This line's a lot longer, so we'll do it again. No, we'll go again on this side.
Oh, me?
Yeah.
Kurt Weingand, Midwestern University, Glendale, Arizona. I just wanted to get some clarity around the regulatory filings under which these studies are conducted. I would assume for human drug development, studies are being filed under the IND. Is that accurate?
Yes, that is accurate. Dr. Layton can correct me if I'm wrong, but my understanding is that in the post-IND setting, they're filed under the IND as a research study.
And there's precedence of that happening and being acceptable to CDER?
Daniel, do you want to comment?
As any IND is open, one updates the IND with either regulatory toxicology studies or new experimental data. And that would also include any adverse events identified in our experience. Exactly what we did. So right, it's the same molecule. You have to inform the agency of all these findings.
Right.
And the timelines and the mechanisms for those disclosures are clear and provided.
Hi, Michael Larimore, UC Davis. Perhaps a slightly different tact. The comprehensive cancer centers have a very interested focus on population sciences, so the environment issue. Maybe, Chan, you could comment on environmental factors or how the dog model may fit in looking at epidemiological factors such as the environment of the human and dog.
Yeah, so I think separate from the issue of translation specifically, I think it's an important area. And Dr. Knapp and her colleagues at Purdue have certainly done a very good job in identifying exposure risks for bladder cancer in dogs, and that has been done by others. Certainly not an area that we've focused really in the area of drug development and translation. And so we've not prioritized those. But if you were to sort of consider an intervention and a prevention strategy, I think this would be a really important area as well. But if I can just ask Dr. Barrett a question. So Dr. Barrett, if I'm sure you've looked at this, so I'll ask you. So, we can look at sort of the better therapeutic areas for success and compare and contrast as a means to get sort of improve our outcomes in cancer. But I was surprised to see that although Lee showed some differences, across the board, drug development, as you pointed out, is hard. It's not just cancer. So then if you look at more successful industries, development sort of industries, it became clear to me that proving semiconductors and devices is much more successful than drugs. And the hypothesis given in this one article that I read, that's all I know, was that simulations was the reason. They can do multiple if-then simulations, and that leads to the success for their industry. Do you have any thoughts on that sort of successful industry versus difficult and hard, and whether you believe that there's something we can learn there?
Yeah, we do a lot of simulations and modeling, both from the biology perspective as well as from the PK, PD perspective. Garbage in, garbage out, you get what you have.
So maybe therein, the dog as a model for simulation could be valuable.
You've been patiently waiting.
Yeah. Hi, Shruti Naik from Mayo Clinic. The question I had was if and how COTC or your groups have engaged the FDA on how they receive and look at the studies from the point of perspective of submitting INDs, et cetera, and might be an easier process for them to understand people who are using this approach. So there's an incentive for us to use the comparative oncology approach.
Yeah, certainly. We've not taken it on from that standpoint. And I do agree that my feeling on this topic is that there is a need for clarity. There will be a session devoted to this topic tomorrow, and Dr. Nissen will be moderating that session. And I think Dr. Layton will be speaking, or part of that panel. And you keep getting called on, so maybe you give your talk today. But I do think it's a critically important question. So I think just clarity and hearing the rationale and reason, I think, would be very helpful to the field.
Yeah. John Layton, I'm in the oncology group at Cedar. So yeah, we'll be talking about all this tomorrow about the IND filing, what we've seen so far, and our regulatory experiences in comparative oncology to date.
So everyone needs to come back tomorrow.
Matthew Frank, Stanford Medicine, Immunology, and the Skippy Frank Fund. I want to thank all the panel for being here, all the members and the speakers. I'm really enjoying this very much. So I wanted to ask a couple of things. One, Chan, on your list, you didn't have immunology as one of the main players, and as we know from the human side, it's been a huge breakthrough at looking at gentler, safer, more effective therapies, and in particular in your interest in metastatic process and going after metastatic disease. And as we look at the potential synergies and diversity and intact immune systems that are closer to ours, and as we think about our companion animals, not just as models, but as patients, and as humans, as both patients and models, as you were talking about before, I think there's some gaps in assays, reagents, biological things, not just genetic sequencing, but if we look for all the antibodies for particular markers, I think there are over 200 CD markers now. I think there's a gap or a funding need for particular specificity in the biologic and the little clues that we would have that would lead to the synergies that would really address our long-term goal of making cancer a gentler way to go and not such a terrible thing to hear as a patient, whether it's a dog, a cat, or a person. Comments from the panel, please.
And I fully agree that the approach could certainly deliver on answers that are not answered in the development of immunotherapeutic approaches, and Dr. Dow has done a lot of this work himself. I do agree that you can cherry pick and develop the reagents you need for every question, or you could sort of conceive of a set of reagents needed to advance the field. And I think both are probably a good way to go. But you're also right that the genomic advances have lowered that bar to develop reagents. And on the protein side, that's still a pretty high bar. And so probably an important gap that needs to be addressed.
Other comments from other panel members? Okay, thank you so much.
Or Dr. Dow, do you want to make any comments?
I think you've addressed it.
Robert Donahue, National Heart, Lung, and Blood Institute. And this sort of follows the previous question, is a lot of the biologics that are being used in the dog are immunogenic. There's differences between the human product and the canine product, and I was just curious to know whether or not there are canine reagents available, or is that in the future? And the second question has to do, again, with cancer therapeutics in the sense of cellular therapies. Clearly, that's exploding right now in the human setting, and I don't see it on the agenda for this particular meeting. Same with tumor vaccines, and I'm curious to know how is that being developed in the canine.
And so I think that then there certainly is no attempt to sort of exclude any therapeutic. There are many that we could have included, and I think in broad sort of themes, cells and vaccines are also very important and have been used in the comparative approach, are being used at the University of Minnesota, important studies in brain tumors. And so they're all, I think, they have an important role. But to answer your first question, so for example, just as in humans develop neutralizing antibody to IL-2, to human IL-2, dogs will develop neutralizing antibodies to human IL-2. And so being aware of that allows you to ask questions in the dog around that understanding.
Well, the same is true of IL-12. I mean, you can go the gamut of the interleukins, and there's going to be some sort of immunogenic effect.
Yeah,
and
but
so
my first point is that it's
is,
no different between the human. The dog is as immunogenic against dog.
Is it because it's a
as
murine
it's a human protein.
it's a human IL-2.
Right.
Right.
There is
or a
no
human
dog IL-2
IL-12.
that's commercially available.
Right, but I would imagine because of amino acid changes, it would be far more immunogenic in the dog than in the human. And so, and I guess glycosylation would come into play as well. So the question is for the biologics, will you need to look at that and take that into account when
Absolutely.
you're utilizing?
Yeah, and so I know about IL-2. So dog IL-2 appears to be a dimer of human IL-2 with similar glycosylation motifs. Every protein is going to be different. There are going to be some proteins that the human doesn't even work in the dog, and there's going to be a strategic rationale for some developing a biological to develop a dog biological as a test molecule for doing these studies that will then result in an understanding of the use of that similar product in the human.
And is that being done?
That has been done, yes.
That has been.
Dr. Richard Vollier, UC Davis. Wanted to ask Beverly and anybody else who care to comment. Basically, the largest explanation for varied drug response is usually pharmacokinetics across the spectrum of all drugs. You showed some very interesting data on the seeding down of tumors to different tissues, the liver, the lung, the brain, where they had different responses to cyclophosphamide. Did they go into depth on looking at that? Was that because of blood supply? Because I would think the next step from pharmacokinetics is actually blood perfusion or tissue perfusion, because the concentration of the drug is the same in the species, but if if the tumor site is more perfused, then I would expect you'd get a better response. Did they see that?
Well, one of the most difficult types of metastasis to treat is in the brain, and that's because the blood-brain barrier.
Right.
A lot of these drugs will not penetrate the blood-brain barrier in very high concentrations. So but the pattern of response there, with the least response in the brain and the liver, is and that was a mammary carcinoma, is very similar to what happens in patients. I mean, when patients get metastasis to liver or brain, those are difficult to treat because, the liver is there to get rid of xenobiotics, and the brain still has a blood-brain barrier. So those are the types of explanations.
Right, unfortunately, we're going to have to cut the discussion short. That was a great discussion. I hope every session is as animated as that, and we'll take a 10-minute break and be back for the next section at 10:20.
That's just a fact.
My goodness, you work hard.